Vivian for hosting it from your place. And it is indeed a big milestone at AIMS University. So our international advisory panel member, Professor Kalaimati, who is here visiting on a working visit at AIMS University. And I just like to inform the audience here, Prof. Kalai is from Herbert Preterm College of Medicine, Biomolecular Sciences Institute, Bioinformatics Research Group from Florida International University, USA. And she's also editor-in-chief for Journal of Medical Microbiology. And just want to inform you all, Prof. Kalai is from, actually hails from Ipoh, and she was educated in a government Tamil school in Buntong, and she obtained her BSc and MSc from University of Malaya. And she went to US and got her PhD and MPH, and she did a postdoctoral in Tufts University in Boston, UT Memphis, and Copenhagen, Denmark. And Prof. Kalai joined FIU in 1999 as a junior faculty. And she also had spent time in Harvard Medical School as a visiting professor. And she's also the founding chair of the Department of Molecular Microbiology and Infectious Diseases at Herbert Preterm College of Medicine, Florida International University. She is a recipient of several prestigious awards, and she has mentored over 150 students from various countries, and in 2011, she was awarded President Council World's Agate Faculty Award as the highest honor at FIU. And she also received the inaugural recipient of New England Biolab's Passion in Science Award for exemplary scientific mentorship and advocacy. And since 1993, Prof. Kalai research team has been working on pathobiology of Pseudomonas, chronic infections, specific emphasis on beta-lactam resistance, alginate overproduction, comparative genomics, alternative therapies, lung ecology, bioinformatics, and nanotechnology. Prof. Kalai shifted her focus towards global health and did a sabbatical in the Pan American Health Organization World under WHO. And Prof. Kalai has over 150 publications, 4 patents, and she has over 120 invited international presentations. And her publications are quite remarkable. And she has published in PNAS, Proceedings of National Academy of Sciences, Annual Review of Microbiology, Nucleic Acid Research, Nano Letters, and so on. Her H index is 29, and it's notable to mention Prof. Kalai was the first FIU faculty member elected to be prestigious fellow of American Academy of Microbiology. Of the 2 Malaysian ever inducted, she is the second and the first woman who were also who was educated in Tamil language, and is a strong supporter and is very proficient in Tamil language. And American Society for Microbiology has listed us as a honorific leadership group. ASM is one of the oldest and largest life science society in the world. So I think I really wanted to read the whole thing. I hope this will inspire the younger generation. And Prof. Kalai is very down to earth and very strong in science. And we are very privileged, and we are having a good time with her and enjoying her thing. I think today's talk will be very relevant. I will please be open. If you have any question, you can feel free to ask. With this little introduction, I would like to invite Prof. Kalai to deliver his talk.
Thank you. Thank you, Prof. Ravi, for the extensive introduction. Really honored to be here and honored to give this talk. And certainly I'd always wanted to visit Ames University. So this certainly has given me opportunity to really get to know your institution and your leadership. And it's really very nice. I really enjoy meeting all the relevant people. So, like Prof. Ravi said, I'm from Ipoh, from a Tamil school, and I do believe that education in your mother tongue really helps you to succeed in science. If anybody tells you otherwise, you shouldn't believe them. I always say in Japan, they learn in Japanese, in China, they learn Chinese, and then they become international scientists because your thinking process changes if you learn in your mother tongue. So it's very different. With that said, I am going to see if I can share my screen. Is there any hearing problem? Does anyone have an issue looking at my video? Vivian, tell me, dear.
Prof, yeah, it's audible.
Can you see my video?
Yes.
Okay, perfect. So for some of you, it's not so brand new, but this is really looking at the phenomena of omics, right? So we are trying to integrate, yes? Oh, slide over there. Why?
It's fine.
No, but I don't see the slide. Oh, there you go. Perfect. All right. Okay, perfect. So I'm going to sort of walk you through some basics and then how we can get a glimpse into the microbiome social network. This is an area definitely we didn't have a chance to talk about it, Prof. Ravi. We should talk about it. So let's look at, okay. This is the brief outline of my talk. I will get into the introduction and microbiome social network, and then I'll get into antibiotics and microbiomes and talk about the dynamism of microbiome and how one could actually estimate how dynamic a particular population in a microbiome by looking at the replication rates. Then we'll get into a new area, which is, just because you see correlation, does it cause? And I'm sure Vivian can tell you all about it because her project was on causation. And the causation project uses a very sophisticated Bayesian network to understand if two things are correlated, are they really correlated, or are they either they caused by something that's common. I will review that definition shortly. A disclaimer. I love using this slide. It says, "Katrathu kaiman nalavu, kalladadu ulagalavu." Simply means that what you know is very little. What you don't know is as big as the world. The whole idea is that we stay humble. We learn from each other. And some of you may know more than I, and together we will teach each other. And I start every lecture of mine with this, even in class. So first of all, we hear about microbiome. And there's always this big confusion that totally irks me, right? Because when you say microbiome, traditionally, we're talking about collection of genomes, right? But when we talk about genomes, they're not live, they are dead material. However, when you say about microbiota, it's a collection of microbes in the environment, right? However, these computer scientists people, they're really responsible for this problem. They couldn't keep it apart. They confused the microbiome, and then they came up with an alternative definition. So microbiome includes the microbes, their genomes, and everything else in the niche. So it's both abiotic and biotic. This is the latest definition of microbiome. And the collection of genomes in an environmental niche is called a metagenome, right? So we know what is the difference between microbiome and microbiota. The second thing is that this microbial community phenome. Well, we just look at the microbes where you can see the parasites and eukaryotic cells and bacteria. When we just look at the genes, the DNA, the gene content and the mutations, we call it metagenome. And not all DNA gets transcribed. So if you transcribe the genome, and you get metatranscriptome, and metatranscriptome, we can study gene regulation, where the particular gene is expressed. But just because we have a transcript doesn't mean there is a protein. So you then look at the metaproteome, that is your translation. And then you can look at the protein content and activity. And of course, now we know metabolomes. These proteins are actually generate metabolites that is important in the life of the microbes in the microbiome. So all these four are what I call the phenome of microbial community. And the phenome of microbial community is really driven by, or the expression driven by the environmental perturbations, the phenotypic traits, and of course in humans, it's the health and disease. Your microbiome will change with your health status. If you're a cancer patient, if you're taking a cancer treatment, you'll kill all the good bacteria, then you also suffer from secondary consequences of microbial dysbiosis, change in microbe. And then we came up with these two new words. One is the metaresistome. In other words, in a microbiome, you do a metagenomics, you get all the genes. That includes all the resistant genes. So if you can collect all the information of resistant genes, we call them metaresistome. In fact, Hovinia Silvaraju worked on this aspect of in her internship. And then we can also determine which bacteria is replicating. Are they static? Are they replicating? So we coined the word Metareplicome. I have to say that I coined it. And the microbial community is extremely dynamic. This is a very old slide that came out in 2012 that showed that microbes are very niche specific. In other words, the oral microbes are very different from urogenital microbes or GI microbes. And of course, this is based on one study. And we know that when it comes to microbes, they are very complex, just like humans. We interact, we communicate, so microbes communicate through signaling molecules. We cooperate, we share resources, we discuss things, how to do, but it can also, bacteria cooperate and share resources. Not all bacteria have to produce all the compounds, half of it can produce. We call them the cheaters, they cheat. They use the other people's resources. So they compete for resources, we all compete for resources, so are bacteria. And we humans will produce goods and services. In the case of microbes, they produce metabolites. And humans will kill with weapons, bacteria and microbes, and they will kill with toxins. So population is very dynamic, right? The human population is equally as dynamic as the microbiome microbiomes. However, in the same study from 2012, this Human Microbiome Project, they looked at 242 people. Not only they found there are specific bacteria that is very specific for a particular part of the body, they always also had a keystone bacteria. What is a keystone? A bacteria, if you remove, the whole system falls apart, right? So we have, for example, in our skin, oral cavity, we have Streptococcus. And then on the skin, we have Staphylococcus. Of course, in the human female vagina, Lactobacillus is important. So this keystone bacteria also drives the health of the microbiome in humans. And how can you disrupt the healthy microbiome? One of the major disruptors of a healthy microbes are antibiotics. And there are two kinds of antibiotics. One is bacteriostatic, the bacteria will stop growing until the antibiotics is siphoned off, then it will start growing. And then the bactericidal is the antibiotics that will kill the bacteria as soon as it hits the bacteria. So there is bacteriostatic and bactericidal. What happens is bacteria doesn't want to die, right? So it tries to develop resistance. It wants to fight the drugs. And that's why we end up with antibiotic resistance, because we haven't learned how to live with them. And when you eat antibiotics, you kill a lot of bacteria, and then the whole composition changes. There are short-term consequences. You get diarrhea. Many of you know, if you take antibiotics, you get diarrhea. In the hospitals, there is Clostridium difficile or Clostrioides difficile associated diarrhea, which is called CDAD, which is a big problem in the hospitals, especially when the patients are given clindamycin, and then Helicobacter pylori can also become an issue. But some communities show resilience and some species never return. This was a very nice paper that looked at the survivors. As soon as you give antibiotics, whole bunch will die, and then some of them will start growing up and coming back. And then there are a whole bunch of opportunities. These are bad bacteria. They go up because they can survive the antibiotics. They come down, but they continue to be in the environment. And then you have the de novo colonizers. There's the bacteria that not necessarily good bacteria, but they start taking over the environment. That's why you have this dysbiosis. And then there are some really good bacteria gets lost completely. They never recover. So a person who takes antibiotics never recovers from this whole microbiome. In fact, there is a whole new theory, therapeutics, where before your cancer treatment, collect the microbiome. And then after cancer treatment, you start taking the pills with the bacteria, right? So the dysbiosis in microbiome is well established. And the question we asked is, can we infer the dynamics of the taxa, right? We talk about yes, no, present, absent. Can we actually say, are these bacteria replicating or not? So in 2015, there's a paper by Preterm et al. He talked about how we can measure replication rate. Since then, there's another paper, it's the codon usage bias. Don't ask me anything about it. That's something I'd like to implement, but that's fairly new, okay? And then we looked at how we can combine multi-omics approach. So can we combine metagenome, metaresistome, and metareplicome to understand the dynamics of the population? So the rationale to find whether particular bacteria replicating is actually come from a work with Vitalis Debliankin, who also was a student host for Vivian and Havinia. They worked with him very closely, and he's a PhD student who came up with the slides. That's eat up on the right. Did you notice that?
It's only the screen. You're okay.
Okay, it's only the screen. Okay. And it's a very simple rationale. If you remember, if bacteria is circular, right? And the diff side is where the termination site is, where the bacterial replication terminates. And OREC is the replication starts. It's a circular, so it's going to go both directions. They end with the diff. So you can then calculate the number of transcripts across this origin of replication versus number of transcripts at the termination site. You can identify peak to trough ratio, and that is a PTR value, peak to trough ratio. You can actually identify which bacteria are actually replicating in the microbiome. And so Vitali implemented this, and this replication rate analysis became very popular last couple of years. And it only works if you know the complete genomes, right? You need to have the start and the end. However, there are new programs that is called metagenomically assembled or de novo assembly. They call the MAGs. You assemble all the genome in the microbiome and you create MAGs. And then you come up with these PTR values. There are several groups and algorithms, and we use BPTR, that's for complete genome. But you can use IRAP, GRID, and DEMIC to do incomplete genomes. So there are many other softwares. And one of the things that when we were teaching the students from AIMS is for them to realize that there's more than one program to do everything. And you really have to look at each one and decide what best works for you, right? So the data set we were using is infant microbiomes and dysbiosis. It's very well known when your infant is born. It's very much sterile. The microbiome in the infant is dictated by the hospital, the mothers, and whether the baby is delivered C-section or vaginal delivery. It's very important for vaginal delivery. Feeding type, because they get the skin microbiome from the mothers, and also the home environment. When you have a term infant, right? In the term infant is somebody gone through the 9 months, they were born, and if they had some issues, and then they have a dysbiosis of microbiome for some reason or other, it can be correlated to colic, or the child cries a lot, atopic disease, or type 1 diabetes. The preterm infants, on the other hand, they are born obviously before the term is over, and as soon as they're born, they're given an antibiotics, and they start observing the child, and they keep giving them antibiotics. One of the major problems we see in preterm infants is they have a lot of dysbiosis associated diseases, particularly necrotizing enterocolitis. Basically, the intestine starts they turn into brown and bloody, and then they lose their intestine. They can have late onset of sepsis if you have enterocolitis or bacteria in the blood, and many of them also have a neurodevelopment impairment. So they do have suffer from mental retardation and etc. Okay. So we use this preterm infant microbiome, and this preterm infant microbiome, we got the abundance data, which is the total metagenome, and then we also calculated the replication rate, that's a Metaresistome, and then we asked the question that whether the replication rate and abundance causes or if there's a correlation, and then we looked at the resistome profile, the Metaresistome, and again asked the question, is there a correlation or causation? In addition, we also added maternal variables and infant variables, because when the children are born, these preterm infants, they're given, external drugs, they're given milk, and all kinds of different things. And then the mothers are treated as well as the infants are treated. And mothers are often treated with antibiotics, so they could contribute to the AMR. So again, the question is there a correlation or causation? Okay. So these are the three questions we try to answer. So this preterm infant gut data set, and this is the nice thing about the bioinformatics students, and I'll tell all the faculty here, is that there are so many data sets not well analyzed. So you don't need to go and create a data set. You just identify data sets that allow you to ask questions and develop or apply some of your methods. And this infant data set, it came from the Gibson's group, and it was published. They had 82 infants, and they were all preterm. So immediately they all given antibiotics because they're preterm. Of that, 49 children were given follow-up antibiotics, beta-lactams, ampicillin, meropenem, tacrolimus clavulinate. Tacrolimus is a beta-lactam, clavulinate is beta-lactamase inhibitor, and they were given macrolide, it is just a gentamicin, and glycopeptide vancomycin. And then they had 35 infants, they did not get any drug after the first drug. So we are comparing the antibiotic cohort and control cohort. And so Vitali developed this pipeline. He took the metagenomic samples. So when you have such a large samples, right, all these samples, how do you process? So he used the project of divide and conquer. So he divided into many different little data sets, and we actually got paid to get some Amazon software and space in the Amazon cloud. And so he could divide all the bacteria into several groups, and then you bring it back after you analyze it, right? And so he used to reference genomes, which had 30,000 reference genomes. That's a lot of reference genome. And then you identify the taxa, and then you calculate the abundance, and you determine the PTR values, and all these were done in this pipeline, okay? And basically, we could identify what species were abundant in the samples, and you can see Klebsiella, E. coli, Enterococcus faecalis, and Physium, they all are bad bacteria, right? Those are called the escape pathogens, which are really not good, even epidermidis. So this is top 25 or 20 highly abundant bacteria. And then the question is, which one of them are highly replicating? Is the really highly high bacteria, in this case is Klebsiella pneumonia, is it also replicating? Yes. But if you look at the Citrobacter, you hardly see here, very little. But you will see in the next slide, it's really replicating a lot more, right? So we can also determine which one is replicating. And the next question to ask is, which antibiotics is causing this bacteria to replicate? Remember, we know what antibiotics were given. So this is the PTR values. So this is replication rate. And then the average is 1.61. When you give these babies gentamicin and vancomycin, nothing happens. If you give them ampicillin and meropenem, you see the replication rate is dropped. That is expected. If you have beta-lactam antibiotics, they simply stop from replicating. And then they'll pick it up after the antibiotic is gone. Which was very interesting is ticaracillin and clavulinate. Ticaracillin and clavulanic, once you mix with beta-lactam and a beta-lactamase inhibitor, your PTR value went up very high. So it's a very significantly high PTR due to treatment of TIM. Okay, ticaracillin clavulanate, the short form is TIM. What it tells us is that this individual should not be given TIM as a treatment, right? The hospital need to know that, whether they know it or not, I don't know. So then we can compare taxa with the PTR values, and here is a big graph. I'm going to pick one and explain to you. Let's take a Klebsiella pneumonia. You can see the abundance with the TIM, it was very high, and you see the PTR is also very high. And you can see it's also significant, right? And the control, you don't see a whole lot, but in the ampicillin and meropenem, they didn't change, right? But, and how about, so in this particular case, high PTR, high abundance. If you look at aerogenes, there is low abundance, high PTR. In other words, this bacteria is in very low to start with. The antibiotics now is helping them to grow faster, and which then it will replace the good bacteria. This is bad bacteria. And so Klebsiella pneumonia is one of the escape pathogens. It is part of the US definition that is very, that we are very scared of in the hospitals. They're responsible for pneumonia, bloodstream infections, wound and surgical infection, and meningitis. But when your patient has Klebsiella aerogenes, we know that the patients have a poor clinical outcome. How it contributes to the poor clinical outcome, we don't know. Now, you saw that PTR values and abundance not necessarily correlates, but what about the resistome profile? So there are 18 different databases, and this is one thing that I was very proud of what Jovinia did. She found more databases than I knew. And one of the things that she put together a talk to tell us what's the difference between all these databases and why you should use CARD over the others. RG is very good. It combines 9 databases, but look at when it is updated. It's not updated continuously. So whereas CARD is monthly updated, so you're likely to have a current, really current data. So now we look at the metagenome samples and we use the bow tie alignment against the CARD database and identify all the resistant genes. And this is the resistome. So we identified about 975 resistant genes. Some of the genes are very highly abundant, like ACRB, MDTC. You can see there are 6 million hits per million read. So this was very high. And then we wanted to ask the question, if there are 975 resistant genes in these infants, how many of them are differentially abundant in the group with the cohort of treatment cohort and the non-treated cohort? We had 320 genes. And then these statistics comes in. We use Mann-Whitty test and Benjamin Hodgback for p-value correction. And then we identified 46 genes that are associated with the group that got the T, which is the ticaracillin clavulanate, and of which 30 were positively correlated with average PTR or replication rate, and then 25 of them were associated with Klebsiella pneumonia. And if you look at what are those genes, you can see these genes fall into various efflux pumps, and many of them actually cause resistance to many drugs, not 1 or 2 drugs. If you have ACR-AB is expressed, you're resistant to all these various drugs. So it's really sad to see these genes are highly upregulated or expressed in these infants' guts, which means that if this infant were to get sick, you definitely don't want to treat them with azithromycin. The child will not respond to it. Okay, so the next question is, we looked at the abundance, we looked at the replication rate to see how dynamics, and then we looked at the antibiotic resistant genes and how abundant they are. The question is the abundance replication rate, and are they caused by them, or is there a cause and effect? And a very good example, we all use different example to explain these, and I know Vivian has a different one. And the idea we say, okay, when it's very hot, you go get your ice cream, right? When the temperature is also very hot, there are people who get angry, there's an increase in homicide rate. So does eating ice cream cause homicide to increase? There's a correlation. But what is the thing is that there is a confounding factor. The factor that causes people to eat ice cream is the same as the factor that make people angry. It's the hot sun, right? So it's a dry, hot sun. So this is what you mean by correlation and causation. This is a big area now because you want to show that there is a causation. There's simply not a correlation. So this work was done by Sazal and Stabliantin. Musfiq Sazal is a PhD student from Bangladesh, and he really came to develop this causation thing with the Bayesian network. And I'll try and explain to you how it works. And then we took it to this study. And we have another paper coming up shortly from Vitaly. Okay. So the Bayesian network is very simple. Every node represents a random variable. And the edges are linked pair of nodes, right? So the edges are associated with conditional probability. Let's look at this example. B affects A. However, the impact on A is the probability of A over B. In other words, what percentage of A actually contributes, B contributes to A, right? So you can actually give a value, right? A quantitative value, but you also have to set some parameters like no cycles, no going back and forth. So then here's another one to look at. If you have many values, B1 to Bk, right? And the effect on A could be B1, Bk could be anything. So you could actually calculate how much B1 contributes to A, how much Bk contributes to T. You can create a table, a matrix, and then you can develop those whole thing. The only limitation with this study is that we ignore time. So you can determine whether a particular link exists or don't exist, whether a particular link causes the disease or not. And we can also infer a magnitude of influence and also identify disease node. Like, for example, if this is a disease, is D, G, and D and G contributes, D and L contributes to disease. So we can actually find the variables that actually contribute to the disease. And we wanted to test that this Bayesian network works, that we really know that it can tell you A leads to B to C. So at this point, Giri, my husband, was asking me about how can we prove that? And I said, it's very well known, the order of bacteria that colonizes in your thing. It's a very old biofilm work, over 30 years. People know which bacteria comes first, which bacteria comes second. So if we can take his concept and proof using this community, then your program is working. So we did that, and you can see we published it in BMC Genomics, and this is also part of his PhD thesis. And so we then took abundance, replication, ABR abundance, and we use this BNLearn-R package, which is for the causation studies. And this uses a particular algorithm called PC stable. There are a few more algorithms besides these, and we still has been exploring that. And then we added various variables, like the infant variables, how old the baby was, what was the birth weight, how much milk or what kind of milk that contributes to this baby. And then in terms of women, she was taking many antibiotics, so we looked at all the antibiotics, the postmenstrual age, etc. And again, we sort of restrict the network. We can't just go in cycles, right? So without going into detail, what we found is that very complex network. We can't figure out what it is, and I hate this network. So I had to create my own, right? So here is an example, Klebsiella pneumoniae, and we know it's highly abundant, but this abundance is driven by these 3 genes. And this ACRA is particularly in individuals with TIN, right? And you can see Klebsiella pneumoniae, remember, high abundance, high PTR, and now we know which are the genes that contributes to high abundance, which are the genes that contribute to high PTR, and we can now go back and see where these particular genes are, right? And then you look at this, you'll see many of them are efflux pumps, which means they're going to kick out drugs like there's no tomorrow. And then if you bring other variables, it's the same figure. Now you see caffeine affects phosphomycin resistant in the babies, and then azithromycin from the mother affects phosA5 that eventually affects the replication rate. So we can literally determine factors that influence abundance or replication rate. And this is more of it in Citrobacter frundi is very well-known bacteria that cause necrotizing enterocolitis. And as we know that these children do suffer from necrotizing enterocolitis, and this is driven by all these various factors. So in conclusion, what we did is we're able to integrate 3 omics data, Metagenome, Metaresistome, and Metareplicome, and we show that this power of multi-omics and the new questions, especially in able to show the causation, right? And we can say, you know, which antibiotics cause resistance or replication increase, which you would never been able to do in a microbiome, because most of the time we do in the lab, we look at the replication, because number of bacteria, yeah, these antibiotics cause this bacteria. So this is very in situ, right? And then because of this causal relationship, we see that cross resistance to unrelated antibiotics, because even though they only gave Kim, but there were other antibiotics that the child cannot take because there are other genes that are present. In fact, they contribute to the replication rate. And we did certainly see the influence of maternal antibiotics and the kind of milk they get. And we were able to integrate the various omics to pathogens and bring it to the point where we show these are the pathogens that are reacting to the presence of antibiotics and contribute eventually to the multi-drug resistance seen in the infants. With that, I want to end in our funding agencies. We have many microbiome projects. In fact, my lab started working on microbiome in 2001 when nobody thought it was viable research and they thought I was crazy. But I hired Dr. Diera Mills, who was a soil biologist, who were looking at soil microbe as a biosensor for the health of microbes. Because when you have the microbes where you have changes because you're giving particular manure or not, you will have a change in the health of the soil. And so with her came Liliana and Sasha and Melissa, and those 3 students tried to use microbiome to separate soil for forensic science, because the soil microbiome of a particular soil is unique. So if you think is Dr. Lee committed a crime, you just take a soil from her shoes, and if she had been to the site where she dumped the body, you can take that microbiome and compare it to her microbiome and show that she was at the crime scene. Okay, it was a very popular paper. We got published in Forensic Science, and then of course we went into vaginal microbiome, lung microbiomes, and mostly vaginal and lung. Bread was patients which were given drugs, and then Natalia worked on some of the verification of some of our data set. And this work would not have been possible without the group of Narasimhan, and Melita Yarik is a student who passed away. Unfortunately, she was hit and run in front of her house, and she developed a good primers to do the 16S RNA. And then Cheng Yong and all these people got PhD from working on that. Daniel, Vitali, Musfik, they all graduated with PhD during pandemic. And then Camilo also tried to express microbiome in a very fancy way, which I still don't understand. And we have a lot of collaborators. So we are a public university. Our medical school don't have a hospital. We share the hospital across the city. So we collaborate with Michael Campos on alpha-1 deficiency and smokers, and Michael Light on cystic fibrosis microbiome, Ziv from Carnegie Mellon on which I didn't get a chance to talk about, is how do you do time warping? Time warping is that the way you will reach to point A is different from the point A she reaches. So there's a difference at the time it takes from go from A to C. So there is a formula, you do a time warping. So you both will reach A, but you do a time warp. It's a very fun stuff. Ziv really did a post sabbatical and they worked on time warping. Dr. Purnima, I work on vaginal microbiomes. Dr. Jim Entry on soil microbiome in US. And Dr. Balachandra Dhanajayan is the soil microbiome from India, Coimbatore in particular. Melanie, she looks at ADHD, so attention deficit disorder. Now we know a lot of the neuropsychiatric degenerative disorders have a strong connection to microbiome. That paper is stalled now, but we will publish. Of course, Trevor is my right-hand person, and he has taken the ADHD project from me. I have his manuscript sitting on my table. And Sabrina is interested in public health microbiomes, how microbiome change in public health. So these are all these various people that over the years they were involved. And with that, I like to thank you. And many of these thank yous don't have nandri, so I had to add one. But with that, I shall end the conversation. They even have Korean and Chinese and all kinds of, even terima kasih is there. So, yes. Okay, I will stop there. That's it. Thank you. How long did I take?
I think it's quite good and very fast and very useful lecture, Prof. I think it's a 45 minutes. Yeah. Yeah.
We can sit side by side with podcast.
I can come that side.
I can move the camera there.
No, no,
Everybody,
it's fine.
everybody can saw the conversation.
Okay, I'll come. But you ask me to sit next to you. This mic is on.
Yeah.
Okay. Prof. Kalai, this is the time to learn.
Okay. Mhm.
And just maybe teach us a little bit on the basics. Actually, we have a lot of undergrad students who are just learning to learn what is origin of replication, what is diff, and so on. And maybe you can give us a little bit fundamentals of Odyssey and the fundamentals of this PTR calculation. Yeah.
PTR. So this is why when we do the program, you have to teach the fundamentals of molecular biology.
Yeah.
Right? The fundamentals is not just what is DNA, RNA, but how the DNA replicates. And because it's a circular chromosome, it always replicates. It's a bidirectional. Right? You need to know 5', 3'. You need to know the polymerases, how they work, how they make more DNA. And then we use that information to say if the bacteria is replicating, then we should have more DNA in the origin of replication. Most people would know termination site or ter site. It's really old. If people are still using ter site, they're not up to date. The ter site was displaced with the diff site before 2008. And in my paper, I talk about diff site. So diff site is basically a series of ter sites put together where the replication ends. So the peak to trough ratio is the peak values is at the origin of replication. And then the how many, let's say if you have 4 pieces of DNA that has REC, you have only 1 piece at the diff site, then you divide by 4/1. This replication is higher at the
this is the DNA level, not at the translation level.
DNA level.
This
Yes.
at the DNA level.
Yes.
If you see from original site until diff means it's the complete.
Yeah, so that's why you need to have a full genome, and it took a while because not all genomes are fully sequenced.
Yeah.
But we used to fully sequence genome in our study, the top genome, and we made sure they're all fully sequenced. But now there are programs that allows you to determine the number of PTRs for not fully assembled genomes. They call them MAGs.
It's basically based on the contigs you have.
Yes, yes.
And
And
the
it will
longest
assemble
contig that has
Ori site.
and what are the signatures in Ori sites?
That's every bacteria has certain signature sequence.
Okay.
It's very specific for a bacteria because That's how it controls its own replication.
Mhm.
So the ori site and the diff site are unique to each bacterial
bacteria.
species. Yeah.
Right.
That's why you need the
So
whole
from
genome.
ori to diff is the original replication until there is one complete cycle or a half cycle?
Half cycle.
Half cycle.
Yeah.
Okay.
So it starts here and then the
The replication is bidirectional
and it ends at
and
the diff
ends
site.
like this. Oh,
Yeah.
interesting. Yeah.
It's the diff site.
It's like,
Yeah, and you can go look up the E. coli diff site with multiple TER sites.
mhm.
So when you do genome sequencing, you've got to determine where's your ori site and diff site.
Right.
And that's very critical for tagging the genome.
The PMB01 and all the other original replication, what you see in the plasmids, they are they different or
Yeah,
same?
yeah. The plasmid replication, origin of replication is very different from the bacteria, right? Otherwise, how can, remember that there are some plasmids can replicate in E. coli.
Yeah.
Because they share some of the machinery. But most plasmids, they can't replicate in one bacteria versus another bacteria because the machinery doesn't recognize the sequence.
Okay. That's also the same thing correlates with the copy number in
Yes.
plasmids?
Yeah. Yeah, yeah. And in plasmids, there's another site called PAR site, PAR, PAR site. You need to check what is the PAR site. So. Yeah, so the reason you have it's to just make sure that every bacteria has a unique system to survive.
Okay. Okay. The next thing is, I think we are very clear about Metaresistome now. Metaresistome is also at the DNA level.
But you can also find at RNA level.
RNA level.
Because which I didn't get to is that you get to which is Povinia's work with her project is that she identified hundreds of genes that are in the Metareplicome, but are they transcribed? Very few.
I think this Metaresistome is an eye opener, particularly people abuse antibiotics.
Mm-hmm.
We can show them
Yeah.
clearly
Yeah.
after antibiotic what happens, what
other bacteria
are growing.
Correct.
So I think it's a very nice. I think if we can make some infographics, I think our interns are listening us. I think we want to. The next pandemic, if it all happens, they claim it's going to be an antibiotic resistance.
Oh yeah, definitely. So I call
So
it an ongoing pandemic.
It's an ongoing pandemic.
Yeah.
So I think we have to create a big awareness on this AMR.
Yeah.
It's a big threat to the society.
Yes, yeah. It's called the forgotten pandemic.
And
the AMR. Yeah.
And the word dysbiosis also, it's a very important. We have to be harmonious. Our microorganism has to be balanced and should be supporting us.
Mm-hmm.
It's like putting a nuclear bomb in your tummy.
Yes.
Antibiotic therapy. It's going to, you might get cured or whatever, but it's going to create chaos there in your gut.
Yes, certainly.
I think that is it's a big. I think if you ask me, I'll keep asking a question. I'll leave it to the audience.
Yes.
Anybody want to ask any questions? I think there's one question there. Did I posted it?
Let me stop share. Okay. Chat. Nobody has posted it. Okay. No post. Okay. Any questions from the audience online?
All the students are there.
Yeah, if they're there, they have turned off the video, which means they're not really there. Hey, Sharvin. Yeah, Praveen is there. Movinder is there. All right. Any questions from you guys?
Sir, it's Vilasini there. I think I have one student working on copy number of plasmids.
Why?
You want to know if you put the transformer plasmid inside a Vibrio. I have a vaccine candidate. It's a live attenuated bacteria. So we want to know the copy number. It's an E. coli, basically an E. coli. It has a coli-1 original replication. So when we transform into Vibrio, it works, but we are not sure about the copy number.
How would you find it?
So we are doing the copy number, the conventional method, and also by real-time PCR.
Yeah, real-time PCR is the easiest.
Yeah, that's what we are trying.
Yeah, because you just use the origin.
Yeah.
Because the origin, the real-time PCR.
That's what we are doing.
This is to wake me up. Thank you. I used to eat these when I go to classes so that I don't sleep.
Other way around.
And in Miami, Memphis, they had this crab apple tree. It's an apple tree with these tiny apples. It's so sour. I'll go before class, pluck a few, sit in class. Every time I feel sleepy. Any questions, Vivian?
That's how.
Okay.
Yeah, these people all I knew from the internship. Jayashri asked me a lot of questions the last time I gave a talk. I do remember Jayashri.
Hi, Prof. Yes, yes, I remember.
Okay, no questions. Any questions from you all?
Just a question.
Mm.
If you're looking at microbes, so CRISPR-Cas9, can we,
No, I can repeat the question.
can we identify a new Cas? So maybe there's different Cas.
So the CRISPR-Cas9 was originally discovered in bacteria, you know that, right? It's considered for the provide immunity of the bacteria to phages. And over time, I had identified so many CRISPR-Cas in Phenome, I just ignored it. I didn't want to bother with it. One thing I can tell you that a lot of people try to use CRISPR-Cas in bacteria. It hasn't worked. Don't know why. And we have one guy called Herbert Schweitzer. He's this tool guru. He makes a lot of different tools for research. Molecular tool guru. He told me he tried very hard to do CRISPR-Cas for bacteria. It seems to work best for the eukaryotic system.
Okay, my question was, can we identify a
New CRISPR-Cas element.
for against this COVID virus?
I don't know the answer. I really don't know the answer. It may be wild goose chase, right? So it's really hard for me to understand and to how we can create. But can we identify new Cas proteins? Of course we can. There are so many millions of bacteria that hasn't sequenced them. We don't have the information. You can find them, but I don't know the answer whether it can be or not. Also, I have to tell you that I started that on Pseudomonas pathogenesis for 20 plus years. And then I was doing microbiome on the side. And now because of the COVID and access to data sets, I completely pivoted to bioinformatics. And it's easy for me, I think I find it easy to teach and train students. But I also don't want to be doing research forever. I also want to enjoy life after helping East.
Yeah, it's all about giving back to the society.
Yes, yeah.
It's the bigger picture.
Yeah. That's my goal.
Prof,
Right.
Prof, I want to ask you some interesting questions.
Or a question.
No,
No, go ahead. Yeah.
Prof, I don't have a question. It's not a question. Some people they go for a fasting and starvation. But how does this fasting affect diet affects?
So it's a very good question. Intermittent fasting is very good for your system, for your microbiome. And those days, our ancestors fasted all the time. You know? At least twice a week they will fast. So you give the system time to recover. It's a very dynamic system, and any food that we don't like right now, I have really disturbed the system. But in general, I'm a disturbed, my system is disturbed. Huh? Yeah, food really affects, especially, in 2 months, let's say, especially I'll use India as an example. If Indian goes to Europe or US, 2 months he doesn't return to India, your microbiome is messed up. You can't eat street food. Okay? But fortunately, by and large, in Malaysia, the water is clean. So I am particular when food is sits around. Temperature is too hot, like Chennai Express. Your chutney, the coconut chutney, everything. In
It's a
India,
good
I will
medium.
never touch any
It's a good
antibiotic.
medium.
So I've never touched it, never gotten sick. I've been to India for 20 some years, sometimes 2-3 times a year. I've gotten food poisoning in Malaysia. And this big party, they had outdoor rojak, and then the next thing I know, I ate rojak. Wow. I had to call the doctor. So the intermittent fasting is really good. Yeah. It stabilizes the microbes. You had a question.
I was fascinated by Pseudomonas bifida.
Yes.
It is. I have a theory. We can create rain using that.
Yeah.
It produces the antifreeze ice nucleating protein.
Right.
It produces ice nucleating protein.
Correct.
And apparently, this bacteria stays in the canopy of the rain tree,
Yes.
the leaves, and so on. So, sometimes if you open a fridge, you keep your vegetable compartment, you keep yogurt or something. At 8 degrees, the temperature will be 8, but it will form a very solid ice on the yogurt, food material.
Really?
So, I think this ice nucleating bacteria has a very good, like Pseudomonas bifida and lot of other species has a very good role in creating rain
Right.
in future.
So the story I say to students is, when I was young, when you go out and come back after a rain, your mother will ask you to shower. Unless the rain was very heavy, then it's okay. Because Pseudomonas is one of the major nucleating bacteria,
Yes.
aeruginosa, right? So when you have only little bit on top of you, it's all dry and you're full of bacteria. But if it showers and pours, it washes off. So, and I think it's not an old wives' tale. It is true that Pseudomonas is in the clouds. It nucleates. And so that's why if you get wet in the rain, you should go take a shower. Which is, of course, with the pandemic, everybody is washing, showering more so than they did in the past.
Yeah, but my theory is in typing, they say always it rains. Possibly the typing leaves will have more ice nucleating bacteria.
Yeah, possible.
That might be causing more rain.
I can take you to
That
the
I don't-
exact spot. I can take you to the exact spot where it always makes great sense.
I can get the sample to FIU, we can do Metagenome.
So metagenomic is very simple. Yeah, there is a group in. This is something we try to work with another university, is to set up a service lab. So the service lab is everybody interested in their metagenome, right? You want to know what is in your system. It's really a money-making system. There's one group is doing that in KL. And Giri tried to set up this company with another person. It didn't work out because the people who came to the table are cold. They're not excited. And AIMS can set up a service system. You provide metagenome. It's like 21andMe, right? They tell you. But you can provide them microbiome. So there is a kit which the person just takes a stool sample and then sends it to the lab. So you create a lab here, you process it, and then do DNA. You don't even, you do DNA extraction and sequence everything. So in the US, there is a company, couple of companies we use. They charge us about $150 per sample. But we get the whole data.
The whole data means you get the whole.
Your microbiome, what you have.
It's a 16S.
No, you do, if you do 16S, you only get bacteria. If you do whole metagenome, you'll get also a resistome.
I was talking to you about the question of waste